To maintain a healthy nervous system, of course, is what everybody would like to have throughout their entire lifespan. There are many ways to achieve this. Many things can go wrong over an entire lifespan, but one of the most important things at a day-to-day moment, or minutes and second-to-second moment, is to maintain control of one's excitability in the nervous system. Now, this will fall to many proteins to try to help, but by far the most ubiquitous, probably preeminent receptor that's involved in this role is that of the GABAA receptor, which in summary really achieves this effect by just enabling the flux of chloride ions across the cell membrane. But what's interested us over quite a few years now is the fact that GABA receptors of themselves are modulated by a number of factors inside the brain. And of these, one of the most potent classes are those represented by the neurosteroids. And that's what I want to explore with you, starting with some work which was about 7 or 8 years ago, the initiation of this whole project, and bringing you right up to date with currently unpublished data. So if we look at a sort of snapshot of a very simplified GABAA inhibitory synapse without any of its intracellular linkages, etc., we can see the role of GABA is designated into 2 parts, that of a synaptic transmitter, relying on activating GABA receptors here, which are made up of alpha, beta, and gamma subunits. And outside synapses, where we have the extrasynaptic receptor populations, again, one can see alphas, betas, and gammas, but also this is the home of the alpha, beta, delta receptor family. Doesn't matter what flavor these receptors come in, they will all be modulated by neurosteroids, and these have 2 basic effects. In nanomolar concentrations, they'll potentiate GABA responses at both classes of receptors, and at micromolar, higher, probably slightly non-physiological concentrations, they have the ability to directly activate the receptor in the absence of any GABA. Now, these neurosteroids are derived from 2 major sources, sex hormone progesterone, and also from the stress hormone deoxycorticosterone. So why would we want to study these receptors and neurosteroids in this way? Well, if you apply neurosteroids to animals or humans, they have a variety of effects which can be therapeutically useful, maybe not this one at the bottom, but the top 3 certainly. And if you have dysfunction in neurosteroid production and neurosteroid levels, then there's a large number of neurological disorders that can occur. This is a truncated list. There's many more that can be seen. We wanted to know, really, the question we posed was, which of these actions of neurosteroids are mediated by GABA receptors? Are all of them involved, or are specific receptors involved? To do that, to address that question, we had really to try to solve where neurosteroids bind to the GABA receptor. So that's where we'll start. Now, I had a very talented postdoc at that time, sadly no longer with us. This is Alistair Hosey, who had quite an interesting idea, and we talked it through, that we could look at GABA receptors, how they respond to neurosteroids such as TH-DOC shown here, causing potentiation of a standard GABA response. But we noted that another receptor in the fly, Drosophila, was pretty insensitive to neurosteroids, as shown here. So after many years of homology modeling, site-directed mutagenesis, electrophysiology, we found that a key residue, a glutamine here, in the alpha subunit was quite important for maintaining neurosteroid potentiation, which if you mutate to a variety of other residues, you completely block this effect. So we thought this might form part of a key neurosteroid binding site. And then went on to see where is this residue. So there are 2 homology models shown here. One on the left-hand side is a sort of membrane plane view of an alpha subunit, 4 transmembrane domains. Here's glutamine, tucked neatly in the transmembrane domain region. Here's a steroid molecule as we docked it in place. And if you take a plan view, a synaptic view, looking down on top of the receptor, here again is glutamine here, and this is our neurosteroid docked in place. We found out other residues as well, which we thought defined the binding site quite nicely. So having discovered this, we then thought we can make use of this genetically by now knocking out the neurosteroid binding site on particular isoforms of the GABAA receptor by just taking alpha-1, for example, or alpha-2, alpha-3, etc., and then see what effects of neurosteroids are lost by deliberately ablating particular neurosteroid binding sites. I'm just going to show you one example of our work so far. We have 2 knock-ins. One is on the alpha-4. That's the one I won't show you because that's still too preliminary at the moment. But the alpha-2 we've got a bit further along. We chose alpha-2 subunits for particular reasons. If you look on the left-hand side here, typical principal cell in the nervous system, alpha-2 has an interesting location, particularly around the axon initial segment. It's almost like a gatekeeper here for controlling excitability, axonal output. So we thought it'd be nice and fun to see what neurosteroids did here. And alpha-2 subunits have in the past been associated with anxiety, depression, pain, addiction, and other diseases. So we thought, again, this would be a good place to see how neurosteroids are affecting such receptors. We did a quick check to make sure the alpha-2 subunit, when you mutate the key glutamine residue, loses all its neurosteroid activity, which it does. And so on this basis, we made a knock-in animal and then decided to analyze that biochemically, electrophysiologically, and also at the behavioural level. So I'll just show you little summary sequences of some of these data. So the first thing, of course, is the housekeeping rule to make sure when you make a knock-in animal that everything else stays apparently normal. In terms of the GABA system, just showing you here some westerns and looking at the levels of expression of different alpha subunits, alpha 1, 2, 3, and 4. We've since done 5 and 6. There didn't seem to be any great perturbation by us making this mutant here where glutamine is replaced with a methionine. This is the only change in this mouse compared to the wild-type mouse. So nothing appeared to be untoward, and the mice in the cage, again, looked relatively normal. However, when we looked at hippocampal recordings, these are taken from CA1, and these are recordings of IPSCs under whole cell voltage clamp in the traditional way, which I'm sure you're very, very familiar with. We looked at their kinetics, and we found that compared to the wild type and the knock-in animal, there was an effect on the iPSC decay. The decays were occurring slightly faster. The rise times also, but no effect on iPSC amplitude. And this is without any addition of drugs or whatever in these brain slices and patch clamp recordings. So it suggested there was a neurosteroid tone on these receptors, on these neurons, which we had removed by taking out the alpha-2 sites. This was a bit of a surprise. We hadn't expected in the hippocampus to have such a big effect for alpha-2, thinking it might just be around the axon initial segment. But it shows that neurosteroid activity on this receptor is fairly key in the duration of inhibition of these iPSCs. We also looked to apply a TH-DOC in concentrations that were relatively meaningful in terms of potentiation of GABA receptors, 50 and 100 nanomolar. For the wild type, adding TH-DOC at these concentrations gave a discernible increase in the duration of the iPSC by about 30% or so. But when we look at the homozygote, the knock-in animals, this effect was largely ablated. Not completely, because remember, in these native neurons, we don't just have alpha-2 that can't bind neurosteroids. We have other alpha-containing GABA receptors that will. And so you will see a potentiation to neurosteroids in the decay here, but only at about 100 nanomolar. So this is, it sort of pushed the dose-response curve on to the right-hand side. So quite a big effect for alpha-2 on inhibitory synaptic transmission. Intriguingly, we thought we should also look at extrasynaptic tonic inhibition in these cells. And if you just glance firstly at the records here, which are current records, they're quite small currents. There's no added GABA here. This is again in slices, so we're measuring really very, very fine current margins of about 0 to 10, 20 picoamps or so. You apply TH-DOC, there's a very slow inward current drift, typically consistent with a tonic inhibition, which you can see reversed by the addition of a GABA antagonist by Kuklin at the end here. When we look at the knock-in animals and compare these to the wild type, we were surprised to see that the tonic inhibition, at least by eye, seems smaller. And when we quantified this from mock treatments with DMSO compared to TH-DOC, you can see for the wild type, there's quite a big increase in tonic inhibition, which is markedly truncated by the alpha-2 knock-in. Diazepam, DZP, shown over here, had minimal effect on the tonic inhibition. So this was quite interesting because we again hadn't expected this. It argued that for tonic inhibition, alpha-2 subunits were playing a role. This is not what we had expected. Furthermore, the fact that diazepam is not doing much to the tonic inhibition suggests it can't be a gamma-2 containing receptor. Gammas are necessary for benzodiazepines to work. So it would argue that we have possibly an alpha-2 beta or an alpha-2 beta delta receptor. Either way, this would be quite unusual and a relatively new entity for an extrasynaptic GABA receptor. So having got this background of electrophysiology and biochemistry, we then turned our attention to some behaviour. And we looked at a variety of tests. I'm just going to show you some associated with anxiety, but we checked depression, we checked pain as well, and I'll mention that as I go through. First up was to look at a light dark box to see how our mice behaved. Typically, a usual wild-type mouse doesn't like light. It will always go back into the dark. And we compared both wild-type and our knock-in mouse line in this way. So you can see time to leave the light zone was much quicker for our knock-in mouse, so they didn't spend much time over here. They were equally inquisitive, it seems, because transfers between light and dark wasn't affected between the two classes of animals. And when we look at the time spent in each of the zones, well, the mice were spending less time in the light when they were the knock-in and a little more time in the dark compared to their wild-type counterparts. And in terms of exploratory activity, it suggests they weren't that sedated at all, because in the light they seemed equally active in exploring the areas of the box. And here in the dark, there was some fall off in the dark zone. So overall, this suggested to us that there might be some sort of anxiety link between the neurosteroids and the alpha-2 subunit. And the fact that we removed neurosteroid binding on the alpha-2 subunit seemed to make the mice more anxious. We tried to corroborate this with another behavioural test. This is the open arm maze. So again, the thinking here is that the mice are not very happy to be out on the open arm, preferring the closed arms for security. And when we look at our data, firstly, without any added TH-DOC, this is just natural basal neurosteroid tone, we can see the time in the open arms is reduced for our knock-in mouse, suggesting they are indeed probably more anxious. Numbers of open arm entries, though, appear to be quite normal, so suggesting motor activity, exploratory activity was not that much affected. If we look at adding now TH-DOC and dosing our animals with a neurosteroid by injection, on the left-hand side, we look at the time in the open arms. We can see that with vehicle and then increasing doses of TH-DOC, the time spent for wild type in the open arms goes up. For the knock-in animal in red, there's a lag. At 10 mg per kg, there's a clear significant difference. They don't tend to spend much time in the open arms, but you can overcome this at higher doses of TH-DOC, presumably by now potentiating the action of other types of GABA receptor. And in terms of open arm entries, interesting result here, which we're not quite sure what this means at the moment, but here there's a profound reduction at 10 mg per kg. But once more, as you go up to 20 mg per kg, this normalises, suggesting their exploration is relatively normal. Now, we also checked for activity on the rotor rod, and by and large, between the wild type and the knock-in mutant mouse, there was really no difference in their profile. So it didn't look as if they had any motor incoordination, etc. So if I summarise this work to date, also there's no effect on depression. There was no effect on pain, I should also add, in terms of other tests. So we think basal neurosteroid levels are modulating synaptic GABA receptors. There's an effect on the IPSC decays. In terms of behaviour, it looks very much as if there's an anxiolytic effect of the neurosteroids, which if we remove the binding from alpha-2, mice become more anxious. And so it sort of backs up really what I think the group in Zurich with benzodiazepines, who took a similar approach of making a knock-in animal that was resistant to benzodiazepines. They also concluded that alpha-2 subunits are involved in anxiety. Difference here is that, of course, this is a natural compound, a naturally innate compound we're talking about, not a benzo that you would take, but this is a neurosteroid that we would all have in our brains, probably performing this anxiolytic role. Now, we were quite happy with that, and everything seemed okay. And then there was a problem. And the problem came at an SFM meeting when somebody approached me, which was quite interesting. I remember the moment very well. And the problem is this. Here are two plan views of the GABA receptor, shown in the way we would think these subunits are organised, beta, alpha, gamma, classic synaptic receptor. I've shown some transmembrane domains on the alpha subunit and also on a beta. They would be similar for all the other subunits. And we had postulated in our original paper 7 or 8 years ago that the potentiation site for neurosteroids was within the alpha subunit, within the transmembrane domains. My 4 fingers are transmembrane domains. This is a neurosteroid. It's totally within the transmembrane domains. But for direct activation of the GABA receptor, we postulated that this occurred between an interface between beta and an alpha subunit. Now, the problem arose because the group in question had used a general anesthetic, which they also thought bound at the same interface. They used a photo-labeled AZ etomidate and labeled the interface, and that's what caused a problem, because their data looked really compelling, I thought, which was difficult. Kind of you to provide some props. So my thumbs are the two neurosteroid residues, but with AZ etomidate in position, these rotated. And with etomidate now balanced in place, the two neurosteroid residues, my thumbs, were a long way apart. They couldn't possibly be engaging in binding neurosteroid here at this site here. So it calls into question this activation site. So that spawned another 5-year study, which is still going on, but I have now some more data to show you. So locating the neurosteroid binding sites, like revisiting what we'd done a long time ago with as I say, postdoc Alistair Hosey. Now, we took a more structural biological approach, aided by help from many labs, and we wanted to crystallize a GABA receptor. That was our desire. Of course, that proved technically difficult, still does. We can't do that. So we managed to make a chimera, a hybrid GABA receptor. And to do this, we took GLIC, which is a family member of the GABA receptor superfamily with glycine and 5-HT3. We had this on the outside as the extracellular domain, and the transmembrane domains were of the GABAA receptor. And this we could express, and we could validate its function. And the idea was that if neurosteroids are binding down here in the neurotransmembrane domains, we could locate the binding site if we could crystallize this entity. And here's neurosteroid structure. So properties of the chimera, important to validate. When you expose them to agonists, in fact, GLIC is driven and activated by protons, not by GABA, of course, because there's no GABA binding site. Here's the GLIC current, very, very slow. If we look at a typical GABAA receptor, alpha-1 beta-3, you see the current rapidly desensitizes. If you look at our chimera, in whatever form, our chimera rapidly desensitizes, suggesting it has the profile of a GABAA receptor, a rapidly desensitizing one. Here are some desensitizing residues that we knew from other studies would cause this effect, and they do the same for the GLIC GABA receptor chimera. It's also blocked by a typical GABA receptor antagonist of the channel, picrotoxin, as shown here. But most important of all, the currents of this chimera were potentiated by a neurosteroid. So the neurosteroid binding site was clearly intact on the chimera. This gave us a lot of confidence to proceed with this construct. So after many years of failure, we eventually came to a structure which we were able to resolve in some decent level of about 3 angstroms, sometimes sub 3 angstroms, sometimes about 3.5. And this is what the structure looked like. So here's GLIC in all its glory, the extracellular domain in green, and the GABA receptor transmembrane domains are shown in blue. Here are plan views up here, and this is a cutaway of this section here, showing the ion channel going right into the plane of the board and surrounded by the M2 alpha helices, which typically line the pore in these types of channels. So we found many things. We looked at alignments with other structures, one of which was the fairly recently published GABAA receptor beta-3 subunit, which was shown in an elegant study by Paul Miller, I'm pleased to see here today, with Radu Ariescu at Oxford. We compared it with GLIC, and our consensus was we could see the so-called activation gate. We could also see deep down into the channel where we thought desensitization occurred by another gate, which we had defined in another paper relatively recently. And all of these 3 structures were thought to be largely in a desensitized conformation. It's very hard to define from a static structure, I think as Mike Edwardson said earlier, function per se, but we generally think this is largely a desensitized structure. So if we look at the sideways view of the ion channel now, this is the top of the ion channel, this is the bottom intracellular part. Desensitization gate is about here, where you get this cuff-type constriction of the receptor. The activation gate's up here, and this is plotted as a pore profile, pore radius against distance through the pore. It maps onto this structure here, and you can see our blue structure is very tightly constrained down here, as is the beta-3, suggesting these structures are captured in a desensitized state. That's good for our crystallography, we think, because the protein is very stable, thermodynamically stable, and therefore better and easier to crystallize. So Just a little bit more before I show you where the steroid is now binding. So we think here's a structure through the ion channel pore. The blue shows electropositivity. We tentatively assigned electron density to ions, shown in green here. This is chloride ions trafficking through the channel. And here's a plain view of the GABA receptor. Here's the chloride ions. And we also identify water molecules tentatively again in the ion channel protein. So where is the steroid binding? The steroid is binding here between subunits, and now across the subunit interface, as you can see in that structure there. It's no longer within the transmembrane pore, the transmembrane domains. It's actually across the true transmembrane domains. There is TH-DOC bound, as I say, across the transmembrane domains. This is the new steroid binding site. And here we go again, similar sort of picture. And once more, again, across the transmembrane domain. So we can see the residues which are involved. We can mutate these residues separately, and all of them will ablate neurosteroid action on this receptor. I won't bother with that. So here's the mutagenesis data showing the actual reduction of neurosteroid action. And the beauty of this is, of course, that now with this new structure here of the neurosteroid, we can accommodate the etomidate binding site just above it, which makes sense because these two ligands interact. Neurosteroids will enhance general anesthesia. So there are three conclusions from this study. The neurosteroid binding site is across the interface. It's no longer two sites. One site must be sufficient and necessary now to explain activation and also potentiation. It accommodates the binding of general anesthetics to the same interface and validates this model as a reasonably good model for studying neurosteroid action. My thanks to people in the lab. Duncan Laverty, who pioneered most of the structural biology, an exceptionally talented PhD student. Salandra Selegiset and Liz Durkin on the behaviour of the neurosteroid alpha-2. And my thanks to all our colleagues who helped hold our hand while we did a lot of learning curve on structural biology in the Pasteur, and of course funders here. Thank you very much.
Excellent, fantastic. Do we have some questions?
Hey Trevor, very nice talk. I'm just wondering with the alpha-2 mice that you made, I don't know how neurosteroids are being released from glial cells or other cells, but could that be being affected as well, in addition to affecting the binding site?
The nearest we've got to trying to quantify that is to do measurements of neurosteroid levels in the intact brain, which we've done painstakingly. We don't see differences between the alpha-2 knock-in and the wild type, so it's not as if they compensated by making more neurosteroid. Good point.
Hi, Trevor. So what does this mean for alpha-4 delta-based GABA receptors for neurosteroid potentiation then if an alpha-2 type receptor is mediating the effect? Is contributing a small amount?
I didn't mean to imply alpha-2 is mediating all of a tonic effect, but it is mediating a substantive part in the hippocampus in CA1.
Yeah.
If you go to the dentates or you go to other areas in the brain with the same animal, alpha-4 appears to be playing a larger role. In the new mouse that we have, which is the equivalent of the alpha-2 but is now an alpha-4 knock-in, we still see effects on tonic inhibition. So what I'm saying is I'm bringing a new receptor player to the table as a potential mediator of tonic inhibition.
So they kind of share the role then?
They share the role then, yeah, that's right. And there's a variation in the relative proportions.
Trevor, this is really nice. I wonder whether you or somebody compare the alpha-2, this mutant mouse. Is there any alpha-1? Because you mentioned alpha-1, alpha-2, they would be in different parts of the brain or different neurons. Right? If you do alpha-1, do you predict, are they going to be similar or different?
We would probably predict they will be different. We think alpha-1 has a different role, but that's purely on conjecture based on the forerunning studies by the Swiss group using benzodiazepines. We've not made an alpha-1 knock-in mouse. We've made only two alpha-2 and alpha-4, deliberately to target synaptic and what we thought would be purely extrasynaptic. That was our thinking. So we've not yet looked at alpha-1.
Okay, then follow-up is for this alpha-2 mutation, is it this alpha-2 then goes out of synaptic so that it becomes more extrasynaptic, or is it like really
So we have evidence that the alpha-2 is put into the membrane extrasynaptically. Then by lateral mobility, it shuttles along to the synapse. But there's a population extrasynaptically, we know, and there's also a population synaptically. So it has a dual role, the alpha-2, synaptic as well as tonic.
Yeah, so but after mutant, is that extrasynaptic is more? So did you...
The trafficking of the alpha-2 mutant doesn't appear to be affected.
It's not changed?
No.
Okay, all right, thank you.